ISU West. I work in the laboratory of Dr. Pamela Marshall. I also work with one of her associates, Dr. Francisco Solis, biophysicist. And as stated previously, the title of my paper is Developing a Luminometer System of Continuous Extracellular Manipulation. So the objective of our research project is to analyze intracellular responses to extracellular fluxes in calcium concentration in budding yeast Saccharomyces cerevisiae. Extracellular calcium gets into the cells through a plasma membrane transporter in yeast and is sequestered in the vacuole. So we want to determine the additional parameters that are involved in contributing to calcium homeostasis in this yeast. And we do this by using transformed yeast cells that are that we've transformed with plasmid from jellyfish, Aquaria victoria, coding for the photoprotein aqua. And we use that light output as a measurement of intracellular calcium concentration. So the objective of our study was to design a luminometer system which could continuously manipulate the calcium concentration around these transformed yeast cells in a way that other luminometers previously were not able to do. So aquarin is a photoprotein. It's non-fluorescent, so it doesn't fluoresce under UV light. Only fluoresces in the presence of calcium. It was first isolated by Osama Shimomura in the 1980s while studying the bioluminescence of Aquaria victoria. The gene has been cloned and it can be expressed in many systems to assay calcium concentrations. This is just a ribbon diagram of the protein. You can see the apoprotein surrounding the prosthetic group in the middle. The prosthetic group is choleanthrazine. It is a member of the luciferin family of organic molecules. So what happens is this protein has 3 EF hand motifs which serve as calcium binding sites. When calcium is bound to these sites, the protein undergoes a conformational change, causing this prosthetic group to be converted into choleantheramide and CO2. As the choleantheramide relaxes back down to ground state, it emits this blue light at 469 nanometers. And we use that blue light as a measurement of calcium concentration inside the cell. This technique, in vivo bioluminescence assay, is an emerging one in the biological sciences. It gives us an ability to look at physiological systems in vivo, which is sort of a novel process. Recently, it's become more affordable. These technologies are very reasonable in price. It's become more common in the biological sciences. Most aquarin assays use a machine previously mentioned called a luminometer. A luminometer is basically just a photomultiplier tube connected to a computer. And most of these luminometers come with an injection system that's capable of doing 1 to 2 pulses of calcium. But it's not able to do more sophisticated pulses such as oscillations because it typically can't pump the calcium or the solution back out or circulate it. So because of its specificity for calcium, aquarin is the photoprotein of choice for looking at calcium concentrations inside the cell in eukaryotes such as yeast and in human cells. Calcium serves as a secondary chemical messenger, as most of us I'm sure are aware. It can enter the cell and cause, it can activate phosphorylation cascades which cause changes in transcription of certain genes. There's an interesting parallel between what yeast do with calcium when it enters into the cytoplasm and what human cardiomyocytes do. The yeast, they sequester the calcium in the vacuole, which is roughly equivalent to the human lysosome. But in cardiomyocytes, the calcium is sequestered in the sarcoplasmic reticulum. And it's kept there because, as we know in the sliding filament model, calcium is an important ligand in muscle contraction. So here's just a diagram that shows these interesting parallels between yeast and human muscles. So here you see calcium entering into the yeast cell through transporters on the surface. It's stored in the vacuole, but free calcium in the cytoplasm can cause these phosphorylation cascades to be activated, which cause transcriptional changes in the nucleus. Here on the right, you see human cardiomyocytes with calcium entering through the T2 tubules, where it diffuses across the membrane through transporters and vascular and it's stored in the sarcoplasmic reticulum, binds to these troponin molecules on the myofilaments, and can also cause transcriptional changes inside the nucleus. Or, I'm sorry, that's mitochondria. All right. So one of the difficulties with using this type of assay is it's very difficult to make sense of your data. There are often multiple variables involved, different protein behaviors and genetic responses to the changes in calcium inside the cell. And in order to investigate all of these other phenomena that are taking place, you often need very expensive technologies. And these technologies can turn researchers off of these kind of questions because they don't have the money to buy them. But the use of inexpensive in vivo bioluminescent assay together with mathematical modeling can add to our knowledge of calcium signaling in eukaryotes by using differential equations and taking an interdisciplinary approach. You can use some of these cheaper methods to investigate these deeper questions that typically people are shy from investigating without the right technology. This is just a picture of the diagram of the apparatus we've been working on. This is most of the hardware. You can see back here the desktop computer. These are signal generators which control these pumps. These pumps then pump in solution, DI water, and calcium chloride buffer into the sample, which is located here. This is the luminometer, it's a previously owned luminometer. We basically bought it just for this little piece right here. That's the photomultiplier tube. And some missing from this picture is a third pump of this type, which we use to pump the solution out. The construction involved taking a standard laboratory luminometer and changing it with custom auxiliary systems, such as the pumping system, which we control remotely through a desktop computer. It was designed in response to an earlier investigation into calcium concentrations that met with several limitations on the available equipment. So previous attempts to construct apparatuses like this that can continuously manipulate the calcium concentration around cells have centered on the use of charge-coupled device digital cameras. These cameras are very good, but they're very expensive. The amount of light involved is very small, it's measured in lumens. And so you need a camera with very, very low background noise and high quantum efficiency to be able to image these reactions. And this can cost you up to $20,000. Our team was given about $2,000 to construct a device that was capable of producing this manipulation and measuring the light output. And so we decided to use a photomultiplier tube from a luminometer and just change the pumping system associated with it. So you can see here, these are the two designs. This is the original design on the left, which originally was going to use a CCD camera. When we found out that we couldn't find one cheap enough that was sensitive enough, we changed our design to include a photomultiplier tube. This is the final design with the sample here up in the top right, and you can see it's much more sophisticated. So here's the final design blown up. I'll go through the pieces. So here is the desktop computer connected to the signal generators, which control the pumps, which pull from the reservoirs of calcium and water, which are pumped into the sample, which is shown here. And then not shown is a third pump here, which pumps the waste out. So the sample itself was something that we had to work hard to design. So what we do is we take the transformed yeast cells and we put them in dialysis tubing. We place those in a standard size test tube, which is compatible with the luminometer. And we use a stopper of our own making, made from silicone and tubing, to close the system. And we pump in the solution through this in tube, and pump it back out through this out, not losing any of our sample because it's in dialysis tubing. So this is a closer view of the luminometer. It's previously owned, as I said, and this is our desktop computer and signal generators. In the construction, we had to do a lot of these components by hand. It was one of the big difficulties, one of the big challenges we faced, including the cables connecting the luminometer to the computer, the cables connecting the signal generators to the pumps had to all be made by hand or refitted from cables that already existed. And that was a difficult challenge, but it worked out. And this is a construction of the stoppers. This is my laboratory assistant, Stephanie Yu. She's mixing up the silicone that we use to pour into this clay mold, which we use to make the stoppers. And we made several dozen of these until we got the design right. If you'd like to see videos of preparation of this sample in the dialysis tubing, we've actually posted several videos on YouTube and have a YouTube channel where anybody on the World Wide Web can see those. And this is some of our sample data. This was obtained just this week. It was the first time we were able to actually get data out of the machine, so the machine is fully functional. This was obtained by putting 400 microliters of suspended cells in dialysis tubing and injecting the sample with 5 molar calcium, 3 pulses, about 5 minutes apart. You can see that the light response is modest. Usually these cells, when they're naked in a photomultiplier tube, jump up into the 10 to the 5th, 10 to the 6th relative light units. Well, this is only at, almost 2,000. But it is significant, and it's a good start. So recently our work was recognized by the ASU News in the Science and Tech section on December 16th of last year. The title of the paper, the article was "To Boldly Go, Fearless Research Focuses on New Investigation Techniques." And that is a picture of my other mentor, Dr. Solis, who's not here today. I'd like to acknowledge the Salt River Project Scholarship, which has been funding my research this semester, as well as the solar program at ASU. And my mentors, Dr. Solis and Marshall, were both supported by ASU New College SRCA grants. Here's a list of my references. And I do have some additional slides if anybody has questions.
We have time for one or two questions.
Jordan, I have a question. I've purchased in an equipping lab both charge couple device instruments and PMT photomultiplier tube instruments. And the latter are often much more expensive, but much better, much more sensitive. What do you think does your home-built PMT-based system, if it gets wind out, performs in the commercially available CCD-based systems?
You want me to compare and contrast the CCD versus the PMT?
Performance-wise, what do you think
Performance-wise, I think,
perform the CCD-based system?
like you said, the PMTs are much more sensitive. They can, detect this amount of light, as I said, measured in lumens, it's absolutely not detectable with the naked eye. And so they can detect, reactions that give off minimal amounts of light, such as these. CCDs, when you get the really good high-end ones, can also be almost as sensitive, but really, if you want to image a reaction, if you want to look at certain organelles or something inside a cell, then a CCD is definitely the way to go. Because with a PMT, you don't know where that light's coming from. You just know that it's giving off the light. You know exactly how much in relative light units, which you can calibrate. But, so if you want pictures, you got to get a camera. But if you're okay with just measuring the amount of light because you're pretty sure where that light is coming from, then I think PMTs are the way to go. And if you get a newer device, they can be expensive. But ours was 20 years old. The technology hasn't really changed that much in those 20 years, and so it's perfectly sufficient for our time. So, Dr. Drew.
So, Jordan, I know you just started sort of collecting some sample data, as you sort of showed there at the end. Can you give us a sense of sort of where this is headed in terms of data collection? What sorts of experiments are you planning to employ for this instrument? And can you evaluate, for example, different yeast mutants that have mutations in the various calcium channels, for example? So, can you tell us a little bit about that?
Sure. Our direction with this is hopefully to, as I said, the yeast are an interesting parallel to human cardiomyocytes, and we want to, eventually, once we have the data acquisition kind of worked out, and we know, the right protocol to get good results, we want to start using manipulating the calcium concentration around human muscle cells, and seeing if we can get them to give us interesting responses that we can study. So that's where we want to go with this. We hope that someday this technology, being able to continuously manipulate the calcium concentration around a cell and watch its response, will help us understand heart physiology a little bit better, maybe even treat some sorts of heart disease. And I'm sorry, what was the second part of your question?
No, I was asking if there were
knockouts,
mutants that were available to
So
study.
in our lab, Dr. Marshall has been studying several knockouts that are related to the calcium or the yeast vacuole-associated proteins, these channels that pump the calcium in and out. And she has some interesting data from previous studies on that, which we will also try to replicate in this machine. We're just not there yet, so but that's soon.
Right.
Do we have time for another question?
No, I think we probably
Okay.
need to move on.
All right.
It's time for King, but thank you.